I'm Tristan from Efficience. You're here in a course given by this association Efficience, who is speaking about biorisk in general. And today we will have a fantastic talk given by Matthew Phillips. So Matthew is studying science and marine biology during his bachelor degrees at the University of Miami in the same institute, and also an MD PhD between 2011 and 2019. During this MD PhD, he did a lot of things. That's impressive. He was a lab teaching assistant, a technology representative at a student government class in 2015, also a co-founder and editor of a journal, a student research journal called Medical IBIS, and also a co-founder, vice president, and president at the Biomedical Drug Student Government. After that, he was 3 years a resident physician in Los Angeles before joining the Massachusetts General Hospital, where he studied as a clinical and research fellow before becoming an attending physician. That's a big career already. So today, Matthew will speak about the subject he's passionate about, which is the climate change and the global warming. So thanks again for being here, Matthew. And the floor is yours. I'll let you present. Thanks.
All right, thank you, Tristan. Awesome introduction. All right, so like you said, I'm Matthew Phillips. I'm coming from Boston, just because everyone's sharing where they're from. So today we'll be talking about climate change and infectious diseases and how those two topics intersect. No disclosures. So a bit of an outline. I'll divide it up based on the type of pathogen and how the different elements of climate change affect the transmission and prevalence of these different pathogens. Forgot to put it here, but at the end, Tristan asked that I talk a little bit about how the physician scientist path works in America and kind of how the career ended up going. So I'll talk a little bit about that at the end as well. I'll try to keep an eye on the chat best I can, but if you have any questions, go ahead and raise your hand. I'll try to answer them along the way, or I can talk about it at the end. All right, so to start things off, everyone's kind of seen this chart by now. Earth has been warming since the Industrial Revolution. Fairly common knowledge, this is the hockey stick graph that the Intergovernmental Panel on Climate Change report, the IPCC report had. Basically, the last 10 years have been the hottest on record. Average temperature between 2011 and 2020 increased by about 1.1 degrees centigrade. And it's projected to continue increasing over the next 100 years. So before we get started talking about infectious diseases, I want to talk a little bit about climate modeling, just because this is where a lot of the data comes from and underlies a lot of the data that I'll be talking about. So this is, again, from the IPCC report. These are different scenarios for how much CO2 is getting released into the atmosphere. We care about how much CO2 is released into the atmosphere because it correlates with the predicted temperature of these different scenarios. And so these different scenarios, you can see the SSP 1.9 through SSP 8.5. So these are different scenarios that take into account social interactions between countries. And so how cooperative they are, how much they work together, as well as inequality. And they also take into account the emissions scenario. You'll see RCP as well. And that's when they just take into account emissions moving forward. And so the two that are the most realistic are the 4.5 and the 7. And that kind of translates into about 1.5 degrees centigrade over the next 20 years, up to about 3.6 degrees over the long term, so over the course of till 2100. In the background of all this, we have sea level change. So sea level change is basically kind of already locked in for the next few hundred years. So you can see a lot of the scenarios don't really change all that much depending on what happens with CO2 emissions. You'll also notice that the first 20 years or so of the climate, of the temperature change is also pretty set. That's because there's a lag time between CO2 emissions and changes in temperature of about a couple of decades. So the changes that we're seeing now are because of CO2 emissions that happened around the turn of the millennia. And so these changes in global temperatures are different in different areas. So the local climates are going to be different and have different effects of climate change. So this is a map of Europe. You can see along the northern area, It will get much warmer, but then it will also get a lot wetter. The blue is increased precipitation. And then in the more southern areas of Europe, Italy, and Spain, you'll see that it becomes a lot drier. And so depending on where you are in the world, there'll be different changes, and that'll drive the different risks of disease that happen in these specific areas. So those are kind of the changes. How are these changes going to affect infectious diseases? So just taking kind of a high-level look at the literature, it seems like there's a lot of different ways. So this is from a systematic review that was done a couple of years ago. Over on the left, you can see these are different aspects of climate change. And then in the middle, you can see the different kinds of diseases that they interact with. This is very busy. It has no business being in a PowerPoint slide, but just kind of the idea being that there's a lot of different ways that climate interacts with infectious diseases. So we'll start with the top here with vector-borne diseases. This is what we know the most about. There's the most data on these diseases. So I'll try to give a little bit of a summary on these. So as of right now, vector-borne diseases are currently increasing. So this isn't any of the modeling stuff. This is current epidemiological trends. Vector-borne diseases are increasing. Over on the left, these are data from the United States. Over on the right are data from worldwide vector-borne disease outbreaks. You can see over the past 30, 40 years, there's been a dramatic increase in the number of vector-borne outbreaks globally. And so vectors are primarily insect vectors. And so you'll see ticks, mosquitoes, midges, some of these other small insects are the ways that these diseases are transmitted. So first we'll start talking a little bit about ticks. So climate interacts with tick behavior in a variety of different ways, which we'll see how that affects diseases coming up. But basically, ticks are a little bit longer lived than other insects. They can live for a couple of years. Over winter time, they bury in the soil. And so if winters are more mild and there aren't as many frost, more of these ticks will survive over the winter. And so you're starting off spring with a larger population. That larger population then creates a larger population of their young, which they can then go out and bite people. The season which the temperature is good for them to go out questing is longer with higher temperatures. And so you have more time that is optimal for them to be going out and biting. And then you have a higher population of them going out and biting. Also, you'll see that the range is increasing as well. So you have this like mixture of perfect storm of different conditions that lead to more tick bites. And every time you get a bite, there's a chance that diseases can spread. So what this looks like in the northeastern United States, this is looking at specifically the deer tick, the Ixodes scapularis. This is the tick that spreads Lyme disease. So between 2000 and 2015, you can see just this massive increase in area where there's a high level of ticks per square mile. And then this translates directly into disease incidence as well. And so this is 1996, looking at the incidence of Lyme disease. And then this is from this past year, or from 2 years ago, 2022. You can see that a lot of the states in the northeastern United States have just been completely engulfed by these cases. And then there's also this encroachment westward as well, over into states like Minnesota and Wisconsin. There's also an increase going up into Canada in it as well as. So this translates into Europe as well. So this is looking forward. So this are projections. And so on the top line, these are the winter months. And so the areas where there's a risk of tick bite during cold winter months. And then on the bottom of the screen, these are summer. This is peak season for ticks and how the range will increase. And so you can see that with the more mild scenarios, let's see if I can use the laser pointer. So with the more mild scenarios, you can see that the tick population the risk of tick bite doesn't increase nearly as much north as with the more extreme scenarios. So these are ones where there aren't a lot of global cooperation to fight climate change and that there's a higher level of emissions. You can see that risks of tick bite increases up here into Britain and up in here into Scandinavia as well. So it's not just the ticks that spread Lyme disease that are spreading. So this is projections looking at multiple ticks that spread different kinds of diseases in North America. Red is all the areas where the population is going to be growing. You can see blue is areas where it's going to be decreasing. And so there are areas that will have less risk of diseases, but then you'll have a lot of areas, particularly in the north and in the west, that'll have higher risks of diseases. And so this is for a variety of diseases, everything from Lyme to babesiosis, tularemia, Q fever. All these diseases have the risk of increasing due to the increasing range and increasing bites and time of activity of ticks. So moving on from ticks to mosquitoes, not just looking at range, but the behavior of mosquitoes actually changes based on temperatures as well. And so as the temperatures increase, there's an increase in the number of bites that mosquitoes do. And the reason for this is because the female mosquitoes require a blood meal for their gestational cycle to complete. And with higher temperatures, they can have multiple cycles over the course of their lifetime rather than just one. So they'll be requiring multiple different blood meals for each of these different broods of mosquitoes. And so that translates into more bites in people, more bites, more chances for diseases to spread. Increasing temperature also increases the number of parasites that can live within the mosquitoes. This is looking at Anopheles mosquitoes and malaria. And you have a higher capacity to transmit diseases with temperatures up to a certain point. Once you start getting into more extreme temperatures, it actually becomes lower diseases just because the mosquitoes are dying off. And so this, as temperatures increase, the range of mosquitoes increases as well. So this is looking at the Aedes aegypti. This is one of the mosquitoes that spreads yellow fever, Zika, dengue, chikungunya. Typically, these are thought of as tropical diseases, but as temperatures increase, the number of months where these mosquitoes can be active and the range of them encroaches northward. So again, kind of bringing back to how this is happening now. So this is back a couple of months ago in Italy. There was an outbreak of dengue. A lot of times dengue is brought in from tropical areas. What made this particular outbreak so notable is how many of the cases were spread within Italy. So these are the mosquitoes that are spreading the disease, particularly the tiger mosquito, albopictus, are spreading the disease within Italy. It's not cases from the tropics. And so the spread is more mimicking the tropics as the weather increases and the temperature increases farther north. So it's not just dengue. This is the mosquito that spreads malaria. Depending on the scenario you look at, you can see that the range where there's months that the mosquitoes can be active are increasing farther and farther north. And so this is, again, something that we are seeing currently. So this is from the United States a couple of summers ago, or last year, I think. There were locally acquired cases of malaria in Florida and Texas, but also as far north as Maryland. So this is areas that are more temperate climates. Again, these are cases that are spread within the United States, not brought from other countries. So switching paths a little bit here, we'll talk a little bit about zoonotic pathogens. Zoonotic infections are spread between humans and other vertebrate animals. So here's a summary of some of the viral zoonotic infection outbreaks that have happened over the past century. You can kind of see eyeballing the dates here along the bottom that they've been becoming more common more recently. And this bears out in the data as well. So this is looking at zoonotic outbreaks globally over the past 30 years, and there's been definitely an increase in these outbreaks. And so climate can affect us in a variety of different ways. So again, this is when vertebrate animals, these are diseases that vertebrate animals can spread to people. And so as the habitat of these animals changes, these animals are going to go to different areas. One of the animals that is very susceptible to this are migratory birds. So this is a study looking at modeling where migratory birds are going to be changed. And so you can kind of look at the map of how the United States is going to change. You can see that areas are becoming wetter, areas are becoming drier. There's kind of a movement of these animals towards the wetter, warmer temperatures. There's also a movement towards higher altitudes as the lower altitude areas and as the areas around the equator become less hospitable as temperatures increase. And so the reason we care about this is because changes in ranges of these animals leads to increased opportunities for viral crossover between species. So this is a study looking at vectors vectors that spread Ebola in Africa, and looking at how in 2070, under different scenarios, these animals can change areas, and then how that change in range could affect viral crossover. And so you can see specifically here in the Horn of Africa, there's an increase in the richness of animals that are capable of spreading Ebola compared to the current area where the Ebola host lived. And with this increase in richness, there's an increase in projected first encounters between animals that currently have Ebola and can spread Ebola and animals that have the potential to have Ebola. And so this crossover between different animals leads to new hosts having Ebola, including things that could possibly interact with humans. And so another kind of just current example of how these changes in habitat can affect human health. So currently in America, we're having a fairly large outbreak of highly pathogenic avian influenza, so H5N1. So over the past couple of years, it's been detected in over 10,000 wild birds in every single state in America. It's also been affecting poultry as well as dairy herds. It's been found in a variety of different mammals. So seals, dolphins, grizzly bears, foxes, minks, and recently it was found in pigs as well. And so all these different mammals are concerning because as a virus spreads more and more in mammal populations, influenza is by its nature a bird virus. As it spreads more in mammals, it gets adapted at spreading in other mammals. Pigs are especially concerning because that's where a lot of flu viruses learn how to spread in people. And so this is something that is kind of continually evolving. I have to update this slide every time I give this talk. So currently, as of this morning, there are 58 confirmed cases in people. So far, there hasn't been person-to-person spread. It's all been animal to person, as far as we know. But it is something that's kind of changing. And so with this current outbreak, they think that some of the destruction in habitat, as well as changes in climate, so this is also interacting with urbanization and human development, changing the way that birds migrate, putting them in contact with other birds and poultry and farm animals in America. All right, so talking a little bit about waterborne pathogens now. So talking about waterborne pathogens, I like talking about Vibrio species because I think they're a really good kind of canary in the coal mine about how climate change can affect waterborne illnesses. So Vibrio, they cause about 80,000 infections in the United States every year. There are three main ones, Vibrio cholera, perihemolyticus, and vulnificus. They're an environmental organism, and so regardless of whether or not people are around or not, they live in coastal waters, and particularly in the Gulf Coast of America. They can cause a couple different diseases, seafood illnesses, as well as wound infections. Patients typically already have some kind of underlying condition, so alcohol use or other chronic diseases. And like I said, it can cause a couple different diseases from GI infections to wound infections, and even sepsis, which has about a 50% mortality. So it's a very serious disease. So as I mentioned, it typically is found in warmer waters. So you can see the majority of cases found down here in the Gulf of Mexico, some in California as well. This is looking at the different months where Vibrio are found. This is in 2014. So you can see that the total confirmed non-foodborne cases here are peaking in June, July. August is kind of the peak for the total confirmed with the foodborne outbreaks. And so you can imagine as climates are increasing, you're going to have increasing water temperatures, and that can increase the risk of Vibrio. And so that's kind of what's been projected moving forward. It's not just temperatures, though. So this is looking at the habitat that's conducive for Vibrio vulnificus with different climate models. So this is taking into account precipitation as well. And so as there's increased runoff and increased freshwater runoff, the salinity of the water is changing. So this is looking at the Chesapeake Bay in Maryland. And so you can see the change here. The darker red colors are areas that are becoming more hospitable to Vibrio, thus increasing the risk of human Vibrio contact and increasing the risk of infections in those areas. This is also in the Chesapeake Bay, basically just looking over the past 40 years at how the temperature of the bay has changed to become more optimal for Vibrio. So over the course of the past 40 years, there's been about a 2-month increase in the number of days that the water is a good temperature for Vibrio to thrive. And so this is 2 more months that are opportunities for Vibrio to come into contact with people and Vibrio to spread infections. So this isn't just something that is in America. This is a study out of the Baltic Sea from 2012, looking at the maximum annual temperature and how that correlates with Vibrio cases. You can see that as the temperature increases, the number of cases per year also increase. And then this is just plotting it over time. You can see that from 1985 to 2015, there's a slow increase in the number of Vibrio cases there in the Baltic Sea. So kind of another way to like trace that in the United States, you can see the farthest north infection with Vibrio that's reported every year. So you can see that back in the 1980s, this was down in the Gulf Coast, but as we've progressed through the past couple decades, up to 2018, you're now getting cases up here in New Jersey. There's been cases in Connecticut and coming on to Massachusetts now as well. Basically, it's been increasing about 50 kilometers per year, tracking farther and farther north, increasing the range of places where there's a potential to spread this infection. So I've thrown these slides just because I like talking about how climate change is a very all-encompassing issue. And so there's a bunch of different ways that climate can affect ecosystems and can thus infect infectious diseases. And so this is a lab study that was looking at Vibrio cholera, and they found that the temperature increases the biofilm production, so how it sticks to, which helps it stick to surfaces, but then also how it sticks to specifically to chitin. Chitin is something that's found in the shells of crustaceans. And so why is this important? Well, it's because copepods have been shown to have a reservoir of cholera. You can see that this is as the temperatures are increasing, the percent of zooplankton that have cholera attached to them increase, and the Vibrio species in general also increase. And the reason this is important from an infectious disease point of view is because every single copepod can concentrate the number of Vibrio cholera cells on it. And so you have 10 to the 4 to 10 to the 6 cells of Vibrio cholera. And so an incidental ingestion of just one copepod is giving you a very large dose. And so the infectious dose of cholera is about 10 to the 3 cells, maybe up to 10 to the 8 cells. And so this is giving you an entire infective dose, whereas in water, the concentration is usually between 10 and 1,000 cells per mil. And so you can imagine this is a lot higher density and can lead to easier time getting an infective dose into people. So this is kind of a model of how complicated it is and how it interacts with the ecosystem. This isn't just something that's hypothetical. So there's been cases of basically runoff of fertilizer causing algal blooms, causing blooms of zooplankton, which are then associated with cholera outbreaks in the Bay of Bengal. So another way waterborne pathogens interact with infectious diseases is through rising sea levels, as well as the increase in extreme weather events. So this chart here is looking at billion-dollar disaster events over the past 40 years. You can see that there's a large increase in the number of events that are causing over $1 billion worth of damage. This interacts a lot with urbanization as well. The tropical cyclone count you can see here in yellow is one of the things that's increasing. And then green here is the severe storm count. So this is also interacting with a lot of water. And so looking at Vibrio again, these storms changed not just the ecology of the Vibrio like we talked about, but they also change who's interacting with the Vibrio. So this is after Hurricane Ian in 2022. This was a hurricane that slammed into the western coast of Florida. And what happened was after there's a landfall, you can see that there's this kind of classic outbreak curve, Vibrio vulnificus and mostly Vibrio vulnificus. And so interesting part of this specific outbreak is it was causing a lot of skin infections and wound infections, but it was very virulent. So there's 36 patients that were hospitalized up to 51 days in the hospital. 8 patients required skin grafts. 3 had to have amputations. And then there were 11 deaths. So this is a case fatality rate of almost 30%, whereas typically with Vibrio vulnificus infections, you'll see about an 18% mortality for wound infections. And so the reason, after looking into kind of the data and trying to figure out why this was causing such an increased fatality rate, is they realized that the median patient age was 80 years old. And so these were older, like retirees living on the western coast of Florida, not the people that would typically be swimming in the ocean, getting their leg cut on a piece of coral, like you would usually hear about with Vibrio infections. These are patients that were impacted by the flooding, leading to wound and skin infections. And because they were already in an immunocompromised and elderly state, the infection had a much worse outcome than we typically see in the patients who are exposed to the ocean. Other types of waterborne infections increase after these storms as well. So this is looking at Legionnaire's disease, Shiga toxin-producing E. coli, and Cryptosporidium, all of which increase after tropical storm events. They don't all increase in the same way, though. Different infections will happen at different intervals after the storm. So this is a study this is where the data from before came from, looking between 96 and 2018 at the incidence of diseases after storms, and then also looking at how long afterwards the increase occurred. And so you can see here for E. coli, you're getting a bump immediately after the storm in terms of the increased exposure. And so this makes sense. So this is the initial flooding causing the sewage to come up. The exposure is much worse. Then you have diseases like Legionnaire's disease peaking after the storm. So Legionnaire's is associated with stagnant water. And so as the water's been sitting there for long periods of time, you're getting more and more people who are exposed to potentially Legionnaire's disease. So this is looking specifically at diarrheal diseases. And one of the themes that you'll see with a lot of climate change and infectious disease research is it's not just the slow change that's causing increase in diseases. It's also the more extreme cases. And so this is looking at 32 different countries, mostly low and middle income countries, for the risk of diarrheal diseases. The higher odds ratio here over 2, this is the higher risk of diarrheal diseases. And you can see here that these areas with high levels of blue don't always have high precipitation. So this is looking at the different precipitation levels. Sometimes it's high, other times it's low as well. And so this kind of highlights the complexity of the interaction between humans, water, and disease. And so what this study showed, and other studies have backed this up as well, is that droughts are associated with diarrheal diseases. Interestingly, this is only really true for dry countries. In more humid countries, the trend is actually the opposite. So as there's increased water, more flooding, there's more disease. In dry countries, when there's less water, there ends up being more disease. And so this is looking at the season of exposure. As I said, in dry countries, there's an increased risk of diarrheal diseases. Wet countries, there is, or wet season, there's a less exposure to diarrheal diseases. And so this has a couple different reasons that or associations that they found. So drought severity and temperature correlated with the cholera outbreaks, and then also population and poverty. So these are more human factors that interact with climate factors and can lead to increase in diseases. This is looking at the water availability and the probability of a cholera outbreak. So as there's less water available for use, there's increased probability of a cholera outbreak. And so thinking about why this is, they kind of created this model. And so you can see climate change is leading to decreased precipitation, which leads to decreased water availability. And so this leads to risky water behavior. And so you can imagine if there's less water available, whatever water is you can access, you're going to want to use to drink and to cook and to use for all these different sources. Whether if you have lots of different options for where the water is, you can choose the cleanest one to go to. So as there's an increased desperation for water, people are using riskier and riskier water, and there's an increased incidence of diseases that are possible there. Additionally, the exposure increases as there's less water, the concentration of bacteria increases. And so you have a higher chance of getting an infective dose when you're drinking those waters. All right, so shifting gears again, moving over to novel pathogens, I want to talk about fungi here. So fungi are extremely common in the environment. There's over 1 million species estimated of fungi, but very few of them actually end up infecting people. And so fungi tend to be saprophytic, so they live on like the floor of the forest, eating decomposing matter. One of the issues with infecting people is fungi usually prefer colder temperatures. And so you have a human inside of us is 37 degrees. And so the warmer temperature just really isn't hospitable for a lot of different fungi. And so that's why a lot of them don't infect people. So this is 300 or so are the ones that are actually human pathogens. And so for this, I want to talk a little bit about Candida auris. It's a really interesting bug. So it was first isolated in Japan in 2009. It's called Candida auris. It was infected. It was isolated from an ear infection, hence auris. Since then, there have been cases in over 40 different countries. It tends to have a very high mortality for fungal infection, so it has a mortality rate up to 60% depending on the study. And it's often resistant to a lot of our antifungal treatments. So the bread and butter antifungal class that we use is called azoles. Basically, all Candida auris are resistant to it. The last line, like one of our strongest antifungal agents is called amphotericin B. And about 1 in 20 are resistant to amphotericin, which is worrisome. And so one of the mysteries with Candida auris was, how is it just popping up in 2009? And so basically scientists kind of wanted to go back and see, are there other Candida infections that people have had before 2009? And we just didn't realize that it was Candida auris. And so they utilized a very large collection. This is a paper that came out like a couple years ago, 5 years ago now, looking at this very large surveillance database full of different Candida species. So they looked at over 20,000 isolates of Candida between 97 and 2016. And what they found was there was only 6 isolates that were, once they were sequenced, showed to be Candida auris. So there's one from 2009 in Germany, and then others from 2013, 2014, and 2015 as well. There really wasn't a lot of stuff from before 2009. Census studies come out. I believe there's one found in South Korea that was like very late 90s. But nothing from before that period of time has been isolated. There's been other efforts looking back as well. And so this is kind of weird. So it's like, how did this infection just pop up out of nowhere in 2009? And so Candida auris is interesting. So a lot of the outbreaks tend to be clonal. And so it has multiple genetically distinct clades. And depending on where the outbreak is, it tends to all be of the same clade. And so you have clades that are more specific to South Asia, Africa, East Asia, South America. And all the infections in the area tend to be the same clade. And so what scientists did is they took a group of isolates from all these different clades and went and did a phylogenetic tree and then kind of did a molecular clock analysis looking back to see where these different clades split from each other. And so what they found was, looking at these isolates from people, they found that in the past 50 years, there's widespread diversification of these different clades. And so they kind of linked this to the use of azoles. Azoles about 50 years ago started being used in farming as well. So there's just a tremendous amount of these antifungals being released into the environment. But the more interesting things that they found were the individual isolates within each clade, all separated up to about 360 years ago. Their most common ancestor was up to 360 years ago. So this is wild because all of these infections are being found after 2009, earliest being found in 2000, around 2000. Then the wilder part is these different clades, looking at the most common ancestor between clades. These are separated by thousands of years. And so up to 25,000 years between the South American clade and the rest of the clades. And so this is wild. And so basically the hypothesis is that climate change led to the emergence of Candida auris. So there's been isolates of Candida auris found in the environment in cooler areas like in marshlands. And so the idea is that as the earth got warmer, these different isolates became more thermotolerant. And as they were able to tolerate higher temperatures, they were able to start jumping into other organisms and causing infections. This is actually a little bit dated. The third step actually might not have happened. The more wild conclusion is that the spread of Candida auris happened a long time ago. These clades are separated by thousands and thousands of years. And so Candida auris spread across the world. And then individually, as the earth was warming, the machinery needed to gain thermotolerance and to become a pathogen was present in all of these different clades. And individually, they all became pathogens over the course of the past 30 years with the rise in temperatures. And so a similar pattern was seen in Sporothrix brasiliensis. So another fungal pathogen that jumped from the environment to cats, now into people. And then several fungal pathogens in amphibians are thought to have the same pattern where warmer temperatures in the environment leads to increased thermotolerance. The ability to survive in higher temperatures allows them to become pathogens and survive in the higher temperatures of other organisms. So a few take-home points from the part of the talk about climate change and infectious diseases. There's a whole variety of different ways that climate change interacts with infectious diseases. There's a variety of different ways that I haven't talked about. Fire can help spread spores from other fungal diseases. The fungal ranges of endemic fungi are changing. And so there's really just the way that climate change is an all-encompassing issue and affects so many different aspects of ecology, creates just numerous different ways that climate change can change the landscape of infectious diseases. And so kind of the main points, climate change is predicted to lead to an increase in vector-borne diseases globally. So this is diseases both from ticks and from other vectors like mosquitoes. It's expanding the range and seasonality of these diseases. So I mean, one of the things that we're taught is that you have a patient coming in the summertime, they have a fever, maybe like low blood counts, they had a exposure to a tick. And so then you start to be thinking, oh, maybe this is a tick-borne disease. Maybe this is anaplasma. Maybe this is babesiosis. These traditional associations might not hold true if the season for these diseases is increasing into the wintertime. And so you have to kind of expand the thought process of, oh, this disease could be popping up in the winter. And this is something we are seeing currently in our hospitals as well. At MGH last winter in February, we had a case of babesiosis. And so there's an expanding range and seasonality of these diseases. Increasing spillover events. And so this is something that's obviously very obvious to the world now after COVID-19. But as the animals change their range, as there's more interaction between these different animals, this leads to a increased chance of viral spillover events. Outbreaks in waterborne diseases after extreme weather events. This also interacts with other things like old infrastructure and old sewage systems that have overflow. So after these large events, these systems become overwhelmed, and there can be increased outbreaks of waterborne diseases. And then the potential for new pathogens to emerge. And so you gave the example of the Candida auris, but also Sporothrix brasiliensis. Like I said, other of these diseases have the potential, once they gain thermotolerance, to become pathogens. And we really aren't sure which ones are have that potential. And so in general, just much of the traditional epidemiology that we were taught in school about these diseases is going to change. And so that's kind of the one of the big themes that I think education, and one of the reasons why I give this talk is I think is important moving forward, is that physicians and researchers and people are really going to have to understand that these are not just summer diseases anymore in terms of vector-borne diseases. These are not just diseases of the tropics like dengue and chikungunya. These are diseases that are going to be spreading and going to have new populations where they could pop up. So I'd like to end on slightly more of like a upbeat note. So what are things that we can do as individuals to help kind of combat climate change? So kind of divide it into four different categories. Individual action, education, reducing emissions from where we work, and then also engaging in the political process. So there's a variety of individual actions you can take to kind of reduce your emissions. Living car-free, buying green energy, plant-based diets, washing clothes in cold water, recycling, like tiny little things. Every little bit helps in terms of reducing the total amount of emissions that are going into the environment. And then education is really important. So scientists, 82%, this is a survey done from British adults in 2020 about who they would generally trust to tell them the truth. Highest on the list, you have doctors, you have engineers, you have teachers, and you have professors and scientists, all with above 80% of the public saying that they would trust for information. So spreading the word and educating people is super important. People's attitudes are shifting. So this is a survey. I was trying to add some more like European data this morning. So 93% of EU citizens believe that climate change is a serious problem. And most agree that greenhouse gas emissions should be reduced and bring the EU climate neutral by 2050. Reducing emissions from the healthcare and research sectors is important. And so when we think about emissions, a lot of times we think of like the smog from cars and actual like gaseous emissions that get released. But basically anything with plastics, anything single use are things that increase the amount of carbon that's being used and then put into the environment. The healthcare sector in the United States is one of the largest emitters. So it causes over 10% of the US greenhouse gas emissions. And so there's a variety of different ways from how we handle waste to how we handle disposables and how much disposables we use that can really impact the amount of the CO2 footprint of the healthcare sector. Research is also another place where there's just a ton of different like waste, especially plastic waste. You can think about how all the pipettes you burn through in a given day. So these are areas where there's a lot of carbon use, a lot of a very large carbon footprint, and one that you can potentially reduce on your own. Finally, engaging in the political process. So this is leftists in the United States. 60% of people believe that Congress should be doing more about it. So people agree there should be a more done. On the right is Claudia Sheinbaum. She was just elected president of Mexico. She's actually a climate scientist, and the IPCC report that I was talking about at the beginning of this talk, she was actually involved in creating that. And so these are just an example of people voting for different politicians who prioritize climate change. And so that's very important just because individual action is great. Larger collective action through governments is even better. All right, so that ends the climate part of the talk. Tristan asked that I talk a little bit about the physician scientist career path in the United States. Tristan, do you want me to answer questions about climate stuff now, or just go ahead and finish all the talking and then we can answer questions at the end?
I think maybe we can keep the question at the end if it's fine for everyone.
Sure, sounds good.
Thanks.
All right, so let's see, there we go. Okay, so I'll be going over just like one version of the physician scientist kind of career pathway. This is the kind of my path to becoming a physician scientist. There are other ways to do this as well, but I'll kind of talk about those as we go. So the general path is first is going to university and a bachelor's degree. It's a 4-year degree. I took time away from school to work as a lab tech for a year. Um, then there's the medical scientist training programs in America. And so these tend to be 8-year programs that are organized kind of like a sandwich. So you do your first 2 years in medical school. So the first 2 years of medical school involve a lot of your didactic coursework. So kind of your general classes on biochemistry, microbiology, and some of the general organ system classes. After your second year, you take your step 1 of your licensing exam, and then you go to graduate school. And so then you'll do the entirety of your PhD. The idea here is that some of the general classes that you took in the first couple of years of medical school would also apply to the biomedical science classes that you would be taking in graduate school. And so that allows you to speed up the process at the beginning of graduate school. In addition, over the summers of year 1 and 2, you're also working in labs and trying to figure out who your PI would be during graduate school. And so that also kind of speeds things up there at the beginning. So then you go and complete your whole graduate school. The average is about 4.5 years. Some people finish sooner, and 3 years. Those are usually people who do epidemiology and have data sets that are already made. Other people, because of various hiccups along the way, can take a lot longer. The longest I heard was 8 years. Basically, their PI left in the middle, and so they had to basically start over. And so that obviously took a much longer time. So after you get your PhD, you defend, and then you go back to years 3 and 4. Years 3 and 4 of medical school are your clinical clerkships. And so this is much less didactic work and more time spent in the hospital. And so as you'll be finishing your PhD, you finish, you defend, and then you'll have to jump back into a different class. And so the people that you'll be joining the class with are people who just finished year 1 and 2. So they're basically a whole new group of people that you'll be joining in with. So after you finish the 4th year of medical school, then you go into residency training. So I mean, my residency training was in internal medicine. So that was a 3-year training program. Longer training programs like neurosurgery. Neurosurgery is an 8-year residency program. There are ones in the middle too. Surgery can be up to 6 years. Peds is the same as medicine, so about 3 years. So yeah, so there's a wide variety of different lengths of time that residency training can be. There's some research time that you can get in residency. After residency, then you can do subspecialty training. So I did infectious diseases, which is 2 years. There are others like gastroenterology, which can be 3 years. You can tack on extra things on top of that. So for GI, you could add in becoming endoscopy, and so do another year at the end of that. And so that can extend a little bit further. And some of the subspecialty training can also have research fellowships attached to them. And so at the end of the clinical training, you aim is to be an attending. End of the research training, goal is to be a PI, although this doesn't necessarily have to be the end goal. So other ways that people can do the physician scientist pathway, not everyone actually gets a PhD. Some people just go through all 4 years of medical school, and then they'll do a more research intense residency or more research intense subspecialty training. And through the training they get there, they'll then try to leverage that to become a PI and start their own lab. So that's the other kind of pathway that some people take to become a physician scientist. So for me, like Tristan said, I did undergraduate at University of Miami. So University of Miami, gorgeous campus. I studied marine biology. The initial plan going in was to study sharks. I wanted to study great white sharks and look at like predators. Obviously, that isn't exactly what happened. Towards the end of my undergraduate, I got involved with a research group that was looking at oceans of human health. And so I got involved in a project looking at basically pathogens in beach sand. And so I really loved the research group. I loved how diverse it was. And so I decided I wanted to study oceans and human health. I did a year as a lab tech working with this group. It was a lot of fun. I got to do a lot of projects that kind of combined marine biology with the more human health aspect of it. My favorite was we studied pilot whales and how MRSA, so methicillin-resistant Staph aureus, spreads between the whales. There's a whale facility where a couple of the whales had sepsis, and then one of the people also developed a MRSA infection. So we went there, we sampled the whales, we sampled the sand, we sampled the volunteers, and basically found that like volunteers were bringing MRSA to the facility. It was living in the water, living in the sand, and then infecting the whales. And so I was trying to combine marine science and the human health aspect that I kind of initially joined Miami looking to do. And so one of my mentors, Laura Fleming, she's actually at University of Exeter now. I talked with her. She was an MD PhD. She kind of convinced me into doing the MD part of it. So I was going to do the PhD. Then I joined the MD PhD program there at the University of Miami. Graduate school, I was in the microbiology and immunology department. Again, I was trying to figure out a way to kind of combine my love for like environmental health and oceans with a more biomedical project. And so I ended up looking at polychlorinated biphenyls or PCBs and how they affect the gut. Basically, PCBs are an environmental pollutant that builds up and that biomagnifies, kind of like DDT does in birds. And so I was able to look at like how PCBs affect the gut and the gut microbiota and the epithelium. And so while wasn't like directly going out into the ocean and getting to sample whales, it was kind of connected, and so I could still kind of stay involved with the field. So I then went to California, did internal medicine. I had a little bit of research time, so I studied Stenotrophomonas, started doing more like antibiotic resistance work, which is where I kind of am now. And then moved to Boston, did infectious diseases, and I'm currently looking at Enterococcus. And so I have a couple different projects with Enterococcus, looking at the biodiversity of Enterococcus in the environment. So these are samples from the Azores, looking at Enterococcus in bugs and insects. And then also looking at antibiotic resistance in Enterococcus and kind of moving forward with that. And so this is one version of it. It's obviously a very long pathway. So this whole thing was about 19-ish years. Like I said, some people skip out on the graduate school part of it, and so that can cut it down to 15 or so years of training to become a physician scientist. But basically, it's really, I would only recommend it for people who really want to do both, who really want to see patients and practice clinical medicine and do research. Otherwise, it kind of sets you back behind your peers. So graduate school, a lot of my friends from graduate school who stayed in research, they are PIs who have their own lab now because they got to skip out on this extra 7 years of training. So they have a head start there. My friends who started medical school with me there started becoming real doctors and attendings right here as I was still in medical school. So it puts you behind if you want to only do one of those careers, but if you want to do both, I do think it's really good experience, and you have to really kind of enjoy it along the way as well. So yeah. So yeah, with that, I am all done. I know it's a little bit earlier than I think, but no one ever complains about things being short. So yeah, with that, I can take any questions.
Thank you so much, Matthew. It was very good. And I think we have time for questions. People can also use the chat. And here we have a class also. If you want to ask, you can come closer, I think. I will let people ask questions. Okay, I have one in the classroom. Here we go.
Hi, thank you for your presentation.
Sure.
What you said about Candida auris made me think about all these viruses and pathogens that are supposed to lie under the permafrost. And I wanted to know, as you didn't mention it, if it's a real issue or if it's just something that we read about in the news. And as you're from the USA, it directly affects the territory of your country with Alaska, I guess. Thank you.
Yeah, thank you for the question. It's a super interesting topic. So I mean, obviously this permafrost has been frozen for thousands and thousands of years. And so there's a couple different aspects of it that I would love to talk about. So the first is just from just the whole pathogen. There's been permafrost that's been encasing, they believe it was reindeer, other animals that have been coming unfrosted in like the Russian region. And so they've had cases of anthrax that have been linked to animals that were in the permafrost. And so with different animals that are in the permafrost, there's a concern that pathogens that aren't really seen nowadays will reemerge and cause new infections. And so this is obviously a concern with the zoonotic infections, so the infections that can spread between animals and people. You have animals that have been frozen for thousands of years. What kind of diseases did they spread around? And are those diseases able to jump into people? So that's a concern. Additionally, they've looked in permafrost, and up to thousands of years ago, they've found genes that are responsible for antibiotic resistance. All antibiotic resistance genes come from the environment. They're bacteria's natural defenses against each other. And they basically can repurpose those to fight off our antibiotics. There's a huge supply of them in the environment, but there's an even larger supply of them in bacteria that are frozen in the permafrost. And so there's some new data coming out looking at antibiotic resistance in organisms that are surrounding permafrost. And they're finding that it doesn't require the antibiotic as a selection pressure. These things are just kind of popping up. One of the concerns is from bacteria in the permafrost leading to it. So yeah, there's a couple different aspects of permafrost that interact with infectious diseases, and a couple different things to watch out for. But I think the anthrax story I thought was especially interesting, just in terms of animals that have been frozen in permafrost coming back and the possible illnesses that could be spread from them.
Thanks. There is also a question in the chat from Esther. Hi, Esther.
Oh, hi, Esther.
Yeah.
I think these pathogens infect more and more people and animals, and more pathogens emerge due to climate change. Do you expect that we might see a drop in their virulence, especially if there's more competition among them? So not necessarily. So I think one of the traps we fall into as people is thinking that the organism's goal is to be a pathogen. Um, uh, this PI down from Florida told me that if we were BHI, we would be amazed at how well adapted bacteria are for growing within us, kind of thing. And so these are traits that bacteria are evolving and pathogens are evolving to survive. And we a lot of times end up just kind of being the side effect of that. And so that's with Candida auris. It's growing in the environment. It needs to grow at higher temperatures, and it can pass over into people. And so the virulence isn't always associated directly with trying to like infect people and trying to spread through like interaction with us. Some of it just spread from them learning how to survive in the environment. And so in that case, specifically, you wouldn't necessarily see a drop in the virulence because of the competition. The competition isn't actually really involved in the development of the virulence. So let's see. So Greet wanted to ask a little out of topic whether our microbiome gets impacted by the climate change and whether the pathological interactions could change there. That's an excellent question, and there isn't a lot of data on it. So that's something I would love to look at in the future. I think it's a very interesting question. Our microbiome is really how we interact with a lot of the world around us. And it's also just so exquisitely malleable and sensitive to our environments. And so that is something I think in the future that will be really interesting to look at, is in terms of as climates change, what happens with the microbiome. I will say that just because of how it interacts with the environment, things like changes in food supply. So if there's less plants because of droughts, there'll be less fibers, and fibers associated with like short chain fatty acids in the microbiome, which are good for our colon cells. And so there's a bunch of different like way, kind of like with the cholera and the copepods. There's a lot of different like secondary and like tertiary effects of climate change that can affect downstream. And so I think the microbiome is really primed to get a lot of those more like tertiary effects. But I mean, the short answer is we don't know. There isn't a lot of data that I'm aware of studying that. Following up on the permafrost question, do you know if there's genomic data available on pathogens found in the permafrost? Yeah, there's a bunch of really interesting studies looking at permafrost. And so I think one of my favorite ones was done, I think it was in Alaska, and they were looking at genes found in permafrost from tens of thousands of years ago. And they were able to find the genes that were like precursors to vancomycin resistance and genes that were kind of like proto-beta-lactamases in these ancient bacteria that haven't been exposed to any antibiotics or any human antibiotics. And so then there's also new species that are getting discovered in ice. Carnobacteria is a evolutionary ancestor of Enterococcus. And so that is an organism that was discovered in permafrost in Antarctica. And so it's a really interesting place where people are using sequencing and finding all these new species, finding precursors and ancient versions of current antibiotic resistance mechanisms that are seen in hospitals today. So permafrost is a very interesting time capsule into the bacterial past. And so, yes, I think it's definitely a very fun area of research.
I have a question myself. So
Yeah.
you showed that some pathogen, I mean, as you know, can survive better with increasing temperature, as you show for Vibrio cholerae. But do we know if the opposite is also true? If increasing temperature can kill some or allow them to survive not as good as they were surviving before?
Yeah. So, I mean, if you remember the paper looking at Ebola spread in Africa, the one that was looking at the different hosts interacting with each other and the probability that there'd be viral spillover events. The ones that I was showing were from RCP 4.5, so one of the middle scenarios with emissions projections. When they got up to the higher emission scenarios, so the ones with increase in greenhouse gases now, they saw a decrease in the richness and a decrease in the spread of the predicted spread of Ebola. And it was because of mass die-offs and extinctions. So it's not necessarily a good reason why the disease isn't spreading, but disease isn't spreading just because of all the animals being dead. So that's one kind of more like morbid way that climate change might decrease diseases in certain situations. Um, the other, I'm blanking on the other one. Oh, with the spread of different vectors for vector-borne diseases, as temperatures get less hospitable to them in the southern parts of the country, of regions of the world, and they migrate further north, those southern areas are becoming disease-free from whatever diseases those vectors are spreading. So that's another way that diseases can go down in certain situations with climate change.
Okay, thanks.
What amplitude do you believe the massive loss of biodiversity, in great part due to climate change, will have on infectious diseases spread? That's a great question. So, as I mentioned, like in the most extreme situation, like it could get better. But I really think that the loss of biodiversity is happening in concert with habitat loss and with these changes in migration pattern. And while on a geological time scale, it's happening very quickly. Throughout like a human lifetime or over the next couple of decades, it's not happening at nearly the scale that I think would really dramatically decrease the risks that I talked about in the talk. So I don't really think the loss of biodiversity is going to affect ID spread in any good way. I will say that like biodiversity, besides being good in and of itself, most of our drugs are all natural, are semi-synthetic or from natural products. And so as we lose biodiversity, we're losing potential sources of new drugs to help fight some of these diseases as they pop up. Africa, working, that's okay. Yes, that's fine. Thank you very much. Thank you very much. Cool.
There's a question coming in the room. You want to? Yeah, yeah.
Yes.
One here also.
Hi again.
Hi.
So your presentation gave us sadly relevant reasons to be worried even more by climate change. But I had a question about vaccines because with COVID, we saw that laboratories around the world were more or less able to collaborate and to have a virus rather not quickly, but I mean quickly enough to stop a worldwide outbreak. I wanted to know what you think about the ability and the technology that now laboratories have around the world to fight diseases. I mean, do we have the weapons even to fight viruses that we never faced before, thanks to the experience we now have? Thank you.
Definitely. So yeah, so I 100% agree. So yeah, so I mean the reports from China initially with SARS-CoV-2 came out in like end of December. I believe the official report was on like New Year's. Within a couple of weeks, the sequence was available online. And so I really think like one of the like few like silver linings of the COVID pandemic was just how the global scientific community was able to work together. So I think that was one of the more inspiring parts of the pandemic. So yeah, so us being able to share the data quickly is amazing. I will say that there's a couple different ways this interacts with global disparities. So even now, I haven't checked this morning yet, but there's a some kind of like flu-like disease in the Democratic Republic of Congo that they're not sure what it is, and they haven't been able to figure out what it is for the past couple of days. And that's just because where the disease is, they don't have the sequencing technology there and don't have the ability to kind of collect the samples needed. And so there's people from the WHO going in to help kind of do some like detective work and figure out what's causing this illness. But the surveillance technology and sequencing technology is only as good as if people have access to it. And so particularly with low and middle income countries, if they don't have access to some of these new technologies, the benefits that they give to fighting diseases and monitoring diseases are going to be lost. The other thing I'll say with vaccines is I didn't include these slides in the talk, but one of the ways that climate change affects infectious diseases is through how it affects people and migration. So if you look at areas of the world that are most at risk of drought, most at risk of things that would lead to mass human migrations, a lot of them are around the equator in the global south. And the areas they move are kind of areas further north as well. Droughts obviously can lead to a lot of like political instability as well, and so that can further drive human migration. And so as humans migrate, global inequality, especially in vaccines and infectious diseases, become more relevant outside of the country that has the inequality. And so with refugee camps from the Syrian refugee crisis in the mid-2010s, there were outbreaks of malaria, or sorry, not malaria, of the measles and other vaccine preventable illnesses. And so that's one of the things, like as the climate changes, I think there's a real imperative to help kind of fight global health inequality because no one's an island anymore. I mean, the reason COVID is able to spread so fast around the world is we're all very connected. And with climate change and population migrations, it'll be very important to try to kind of increase equity of healthcare globally to help kind of prevent, to obviously for the inherent good of helping people's health, but also to help prevent diseases from occurring in other places as well. All right, so it looks like there's a few questions. All right. The progressive increase on Earth's temperature and climate change are target as the increase of waterborne and reappearance of diseases outbreak. And and political measures aren't always taking regarding control. How, as scientists, do you things, or how should we be acting? As most recommendations by scientists aren't taken into account. Yeah, so I mean, it can be frustrating with people not listening to scientists all this time. I think, like I said, like we are still, I mean, the data that I showed was actually like before the pandemic, but everything I've seen from stuff after the pandemic, cuts has gone down, but it's still higher from scientists and people in the academic fields kind of thing. And so education remains like incredibly important. I think a lot of this will get better with the changing demographics of governments. Um, if you look at like data on different like generations, millennials and Gen Z tend, regardless of political persuasion, tend to rank climate change as a higher priority of government action. And so as some of the demographics shift and as younger um, people start running governments, I do think climate change will become a more active place where governments are trying to solve problems, regardless of any kind of left-right bent of those governments. But yeah, I mean, spreading the word. And I think one of the things that I like about talking about infectious diseases and climate change is a lot of times when people think about climate change, they think about like the image of like a sad polar bear on a melting iceberg in Antarctica. And that's very sad and provides a great visual, but it's also not super relevant to my life here in Boston or here wherever. And infectious diseases are changing now. And so we're seeing the increase in vector-borne diseases like now. We're seeing malaria in America now. The dengue outbreak in Italy is happening like a month ago. And so these are infectious diseases that are affecting our health in our own backyards right now. And so I think infectious diseases provides a very good venue for discussing climate change just because of how much it interacts with your daily life. I mean, I think one of the places where I start like thinking about like public health discussions with people who disagree with me is I think everyone wants to not die. And so that's like a starting place that regardless of your political persuasion, you can kind of build off of. And so with health, it's like right there. And so like even if there's disagreements, trying to improve health is kind of a more universal goal. And so I think health is a very good way of discussing climate change in a way that just is very relevant to people and people care about. And also it gives people a way to like provide policies that don't necessarily have climate change in the words kind of thing. So like some of the fungal diseases in California, the legislation about providing funding and awareness of those are coming from people who don't believe in climate change. But it's because this disease is prevalent in their community. They're seeing the increase in the disease. And so it's becoming something that needs to be acted upon. And so that's where I think kind of the infectious disease angle on climate change has a couple benefits in terms of inspiring action. All right. How are emerging diseases in the Sahara linked to climate change, and what strategies are in place to combat them? Dr. Fassel, is there a specific disease that you're referring to? I'm unsure exactly what disease in the Sahara the question is about. There are several like emerging diseases that do have climate change links to them. So there's a recent outbreak of the Oropouche virus in Puerto Rico. So that's a virus that is kind of associated with Brazil. It's associated with little midges. But there's been an increasing amount of that virus spreading in more northern places recently. Another virus was just discovered in China, the wetland virus, so spread through ticks. And so there are some of these emerging viruses, emerging diseases coming from vectors. And vectors, like we talked about earlier, are very susceptible to changes in climate and changes in range. So they can change the prevalence of diseases. Thanks for the answer. The loss of biodiversity could even have a negative impact as there are less niche-like barriers to transmission, more biological genius. Yeah, no, that's an excellent point, Nicholas. I think that's something that's absolutely very plausible in terms of losing some of the biodiversity leading to increase in disease spread. Definitely spread of farming and especially like more like factory farming practices can have really negative impacts on diseases. You have a huge population in a very small area that's all really homogeneous, like you're talking about, in this case being livestock. And so you have things like influenza that can start an infection, just kind of work their way through the entire group of animals very quickly, picking up, and it allows it to really optimize for spread in those animals, which is kind of what we're seeing now with some of the bird flu. All right.
I think we have one last question around here. Someone is coming forward again. Let me got a burning question.
Hello, thanks. You talked about the fact that the changement in the climate changed the viruses, but does it also change our capacity to react to the infections, like the immune system or something like this?
Excellent question. So yeah, so there's a group out of Yale who does a lot of really interesting research on this, the Iwasaki lab out of Yale. And so they've done a lot of work with the immune system and viruses and how different changes in the climate can affect the ability to fight viruses. And so I mean, one of the things they found is that if it gets, if the humidity goes down in the air, we lose some of our ability to clear viruses, and there becomes kind of breaks in our mucosal membranes that increase the ability of viruses to get into our system. And so that's one example of how the outside weather can affect the spread of diseases based entirely off of things that are happening within our own body. Colder weather has been associated with like a less robust immune response, possibly one of the reasons why you see certain viral illnesses getting worse in wintertime. And so as there are changes in the environment, we're part of the environment. We're also animals that are part of the ecosystem. And so changes with us will also definitely be affecting diseases, some for the better, some for the worse. And so, yeah, so definitely our own immune system is not excluded from changes in from the environment and from climate change.
Well, thanks everyone. I think it's time to close this session. Thanks for everybody who was here in the room and online. And thank you so much, Matthew. It was an amazing talk.